Hi everyone, welcome to my channel Swarna Ma'am's Biology. In this video, I will be explaining you about the different blood groups present in the human beings along with blood transfusion, lymph and a small note on coagulation of blood. So let us start. In our last video, we have discussed about the different components of the blood, that is different blood cells which are present in the human beings. So here we will be discussing about the different blood groups which are present in the man. So did you see the picture Amar, Akbar, Anthony? Even in this picture, the same blood group between these persons helped them to identify their siblings along with their mother. So blood groups are that much essential. Not only they are essential for the identification of the family members, the identification of the blood group is very much essential during the transfusion of the blood, because incompatible blood transfusion will lead to the death of the person. Okay, so blood groups are very much essential during the transfusion. So when we see about ABO blood grouping, Carl Landsteiner was the first scientist who first identified the different types of the blood groups of the man. He identified based on his experiments. He used it to conduct experiments by transferring the blood between the two healthy individuals. So in some cases, he used it to observe very drastic reactions which used to occur in the recipient's blood, which even led to the death of the person. Later, he identified the drastic reactions or the deaths are due to the surface markers, or which he called them as antigens, which are present on the surface of the RBC of that person, and even the antibodies which are produced in response to that antigen which he gained from the donor. So here we are coming across the words which are called as antigen and antibody. Usually, what is an antigen? So any foreign particle which is not our body, we call it as antigen. Which is not belonging to our body, we call it as antigen. And whenever any foreign particle enters into our body responds to that and produces what are called as antibodies. So whenever antigen enters, our body responds to that to produce antibody. But it is not in the case of the blood grouping. So these antigens are like surface markers which are present on the surface of our RBC. Means these are like markers. If the blood of that person contains A antigen, then we will be naming that person as A group person. If the blood group or the blood cell contains B antigen, we will be naming that person as B group person. Means here in the ABO blood grouping, so antigens are like surface markers which are present on the surface of the RBC of that person. Then what about antibodies here? These are the proteins which are present in the plasma, which are produced in response to the antigen, but it is not against the antigen which is present on the surface of the RBC. Here in the case of ABO blood grouping, this antigen which is present on the surface is recognized as its own body particle. So our body will not recognize it as foreign particle, so our body will not produce antibody in response to that. Okay, so this A group person doesn't contain A antibody, he contain only A antigen. And B group person has B antigen, but his body doesn't contain B antibody. Why? Because his body thinks B antigen is his own, so his body will not produce B antibody. Okay, so here we came across the word A antigen or antigen and antibody. Okay, here in the blood grouping, antigen is a surface marker which is present on the surface of the RBC, and antibodies are the plasma proteins which are produced in response to that antigen. So now coming to the different blood group. So we know what is an antigen and what is an antibody. So if here, if A antigen is present, so I told you that antigens are present on the surface of the RBC. Yes. So if antigens are present on the surface of the RBC, which antigen is present on the surface of the RBC? Based on that, we are naming the group of the person. So when we see here again, So here we will see, we are seeing here A antigen is present. So when A antigen is present, then he belongs to A group person. This A group person has A antigen. His body had studied the structure of A antigen. So his body will not respond to that, will not produce A antibody. But when he gets the blood, or when he is exposed to the B group person or B blood, then his body responds to the B antigen and produces B antibody. So in general, how we can say this A group person contain A antigen, and he will be having in nature, naturally, B antibodies, but he will not contain A antibodies. Then coming to the B group person, his RBC on the surface contain B antigen, but he will not contain B antibody. Why? Because his body had studied about B antigen. So in plasma, he will be containing A antibody. Okay. So coming to the AB group person. The surface of the RBC of AB group person contain both the antigens, that is A antigen as well as B antigen. So this person's body had already studied those two antigen. So it will not produce antibodies against any other. If he gets the blood from another A group person also, this person knows what is A. So he will not produce the antibody, that is A antibody. If he gets the blood from another B group person also, this person already know what is B antigen. So this person's body will not respond to that and produces antibody. So the AB group person plasma usually doesn't produce any antibody. Okay. So what about this O group person? This O group person surface doesn't contain any antigen. So he doesn't know what is A antigen and what is B antigen. So whenever he is exposed to another blood group, his body immediately reacts to produce both A antibodies as well as B antibodies. Usually in nature, in man, we are having A group person, B group person, AB group person, and O group person. When A antigen is present, that person is said to be A group person. When B antigen is present, the person is named as B group person. When both the antigens are present, then he is AB group person. When no antigen is present, he is O group person. Okay. Now coming to which type of the blood this person can give, to whom he can give and from whom he can take. So when we see that, that is this A group person. So when we are seeing about a type A person, so we know that this A group person contain A antigen. So this A group person can get the blood from another A group person. Why? Because this person already know what is A antigen. So his body or blood will not respond to that and produce A antibody. And when he can get the blood from another O group person also. Why? Because this O group person doesn't contain any antigen. So his body, his body will not produce antibodies against that antigen. So we can say that this A group person can get the blood from another A group person or another O group person, and he cannot get the blood from B group person and another AB group person. Why? Because his body is now seeing another antigen B or AB. In AB he is seeing B. Immediately his body responds to produce what are called as antibodies. So you can ask me, what happens if antibodies are produced? When antibodies are produced in response to that antigen, what happens? This antigen and antibody react and produce what is called as agglutination, and produces what is called as agglutination. What happens if the body reacts and produces and forms agglutination? Agglutination is nothing but clumping. What happens when the blood is clumped? It stops the blood flow, results in the death of the person. So agglutination or clumping will result in the death of the person. So that is A group person who has A antigen has to get blood from another A group person or O group person. Why? Because his body will not produce the antibodies in response to that antigens. Okay, so now again we'll discuss about B group person. What happened to this B group person? Already his body contains B antigen on the surface of the RBC. So his body, whenever he is exposed to other antigen, when he gets the blood from another group person, what happens? His body produces B antibodies. Sorry, A antibodies. So in the case of type B person, already his body has B antigen. So whenever he gets the blood from B group person, it is okay. Why? Because his body already have studied what is B antigen. So it will not respond to that and produces antibodies. And O group person is also okay. Why? Because this O group person doesn't contain any antigen. Okay, so his body will not produce any antibodies. But when he gets the blood from A group person or AB group person, immediately his body reacts and produces what are called as anti-A antibodies. So whenever he gets the blood from A group person, it is new to him. So his body immediately produces A antibodies against to fight against that antigen. And whenever he gets the blood from AB group person, again he is seeing here what A. So again A antibodies are produced. So the body fights against that, means it will not accept that. So whenever he gets the blood from A group person or AB group person, A antibodies are produced and causes clumping or agglutination, resulting in the death of the person. So what happened in the AB group person? So in the AB group person, if we see, he contains both the type of antigen. A antigen he is containing, is also containing, that is B antigen also. But his plasma doesn't contain any antibody. Why? Because both the antigen he is thinking his own. So he will not have any antibody. So from where he can get A group person also okay. Why? Because his plasma doesn't produce any antibody. Why? Because this person's body has studied what is A antigen and doesn't produce any antibody. B group person is also okay. Why? Because he knows, he already studied what is B antigen and his body will not produce B antibody. A B group person also okay. And even O group person also okay. Why? Because he doesn't contain any antigen. Okay. So this AB group person can get the blood from A group person, B group person, AB group person, everybody. So we call them as universal recipient. So now coming to the O group person, we know that the O group person doesn't contain any antigens. And whomever he gets the blood, he thinks it as new. If he gets the blood from A group person, he will think that another new antigen entered into our body and produces A antibodies. And if he get the blood from B group person, again he thinks it is new, again produces B antibodies. Again AB, fully his body reacts and produces both the type of antibodies. So he can get the blood from only another O group person only, but Papam, he can give the blood to all the other group persons. So we say that O group person is a universal donor where he can give blood to all the other group person, but he can get the blood from only another O group person. Okay, up to now is it clear? So again coming to ABO blood grouping. So we have seen four types of the blood group are there: A, B, AB, and O, depending upon the type of the antigen which is present on the surface of the RBC. If A antigen is present, then it is A group. If B antigen is present, it is B group. If AB is present, it is AB group. If no antigen is present, it is O group. And we have seen if the B group person is a recipient, he has to get the blood from another B group and O group. If he gets the blood from another AB group, it causes agglutination, what we are calling it as clumping. Clumping means like curd, how it will be clumped, so it will prevent the blood flow, like clumps. If a blood forms clumps, what happens? The free blood flow will be affected, resulting in the death of the person, what we call in our words as agglutination. Okay, so here again we are seeing if the recipient is A, he has to get the blood from A group or another O group. If the recipient is AB, he can get the blood from A, B, AB, is even O. If the recipient is B, he can get the blood from B, O. If the recipient is AB, from everybody, and if the recipient is O, he can get blood from only another O group person. So up to now it is clear. So the same blood transfusion we are showing in the slides, okay, for whom the blood has to be transferred. So Steiner, after he recognized A antigen, B antigen, when there are no antigens, so he named A group, B group, AB group, and O group. He even identified that even though there is a means when he is following some experiments, he was very careful that he was transferring the blood between the same group persons. Even though he is that much careful, he observed some reactions which are occurring, which are again drastic. So later he identified it is due to a factor which is called as RH factor, which is called as RH factor, which is responsible for these reactions. So, what is this RH factor? So, we know that on the surface of the RBC, if there is A antigen, he belong to A group person. If there is B antigen, then he belong to B group person. If there is both the antigen, if they are both antigen, then he is AB group person. If there are no antigen, he is O group person. So we know AB, A, B, and O, depending upon which type of antigen. Along with this, he identified on the surface of the RBC, if there is D antigen, then he belonged to positive. What we are calling it as RH positive person. Means when his blood, that is a blood cell, contain D antigen on the surface, then he is then he is RH positive. When he doesn't contain D antigen, then he is RH negative. Okay, so these RH positive and negative are because of the D antigen along with the respective antigen. For example, here you can see in the case of A, there is A antigen, so he is A group person. And along with that, if he is having D antigen, then he is A positive. If he is not having the D antigen, then he is A negative. And on the surface of the RBC, if he has a B antigen, he is B group. And along with B, if he has a D antigen, then he is B positive. And if he doesn't have the D antigen, then he is B negative. Along with O. And on the O, there are no antigens, we know. But if there is D antigen, then he is O positive. And if there is no D antigen, then he is O negative. On the AB group person, blood, what happens? There is A antigen, B antigen, and even D antigen. So what we call, when there is D antigen, then he belongs to RH positive. When there is no D antigen, then he is D negative. So why we are calling it as RH? It is first identified in the monkey, which is called as rhesus monkey. So we are calling it as rhesus factor, that is depending on the presence of antigen D. We are calling it as positive or negative. So what is the necessity of studying all these? So there is a thing which is called as erythroblastosis foetalis. So this is a most dangerous thing which is related to the RH factor. So we have seen the RH factor, that is positive, negative, that is if the anti-D is present, they are termed as positive. If the anti-D is absent, they are negative. So usually in nature, positive is dominant when compared to that of negative. So when we combine a positive with negative, usually in nature favors normally that is dominant individuals, usually positive will be the outcome. So here, what is erythroblastosis foetalis means? It is the death of the fetus. So how it occurs? When it occurs means when an RH negative mother is married to a RH positive father. Means when the mother is RH negative and father is RH positive, then what about the baby? In nature, we know that dominant thing will be favored. So the baby, if it is Means it is also a chance, probability. Normally, the babies will be having the blood of the mother, but nature favors sometimes the dominant thing. So, if the baby is Rh positive, then what happens? So, this baby, which is Rh positive, in the first pregnancy, is not danger to the baby as well as mother. But what happened to the second pregnancy? So this baby is having Rh positive, means it is having what? Antigen D in it. So when the antigen D is present in the baby, so this can't means in response to that, the mother, there is a mother produces anti D. But these immunoglobulins which are produced are of IgM type. Okay, during the first pregnancy, I'm telling, these will not cross the placental barrier. Why? Because IgM means when we see the immunoglobulins, that is antibodies, IgM is the largest immunoglobulin which can't cross the placenta. So it will not cross the placental barrier and enter into the fetus and destroy that. So first baby is safe, and even mother is also safe. Okay, so this baby, if it is Rh positive, so first birth is safe. Even though the mother reacted to that also, IgM antibodies are produced in her body, but it is not affecting the fetus. So what happened to the next pregnancy? So whenever she become pregnant for the next time, if it is again Rh positive, then this baby will be having what? Antigen D in it. Okay, so already mother have what? Anti D in the form of IgM. But during the second time, again the mother is exposed. So now the body starts producing even IgG type. So what happens? The IgG antibodies cross the placental barrier of the mother and enters into the fetus. That is second fetus. Okay. So now what happens when the so now what is happening? The anti-D antibodies, which are IgM type, now they are crossing the placental barrier and are entering the fetus and are destroying the fetal blood. Okay, so here there is occurring the destruction of the fetus. Usually when we studied about the formation of RBC, we know that RBCs are formed from the red bone marrow. Means there are several stages during the formation of RBC. First, RBC, that is, they will be from the red bone marrow, reticulocyte cell will be formed, and from there, erythroblast will be formed. From there, erythrocyte will be formed. Okay, the maturation, after maturation, erythrocyte will be formed. But here, whenever this that is whenever this Rh negative mother is carrying an Rh positive fetus for the second time, what is happening? This that is erythroblastosis, that is the red blood cell in the erythroblast side will be affected. Means it is not even coming to the erythrocyte stage. Okay, so what is happening here? I will clear explain. So here antigen, there is a immunoglobulin G, which are produced from the mother, are causing the placental barrier and is destroying the fetal blood in the erythroblast stage itself. Okay, so here the fetal blood is affected in the erythroblast stage itself. So we are calling it as erythroblastosis fetalis. Okay, so here what is happening? When the mother, that is Rh negative mother, is carrying an Rh positive fetus for the second time or third time or fourth time, already that mother is sensitized to the antigen D. Why? Because in the first pregnancy it has seen already. So her body knows what is antigen D, and it produced anti D in response to that antigen, but in the IgM type. In the first pregnancy, it is not dangerous. Why? Because this IgM which is produced is not crossing the placenta. But what happens during the second pregnancy? This mother is producing IgG, that anti-D in the form of IgG. So this IgG is a small antibody which immediately cross the placenta. So what happened if it is crossing the placenta? It immediately, that antibodies are attacking the RBCs. See here, they are attacking the RBCs of that fetus. So when they attack the RBCs, they are destroying the RBCs in the erythroblast stage itself. So what happens if the RBCs are destroyed? It causes the death of the fetus. Okay, it causes the death of the blood cells. So what happens when the death of the blood cells? It leads to the death of the baby itself. So it leads to the stillbirth. Some and even before also, it leads to the release of bilirubin and bilirubirin, which are the bile pigments where the liver is seriously affected. And even though even if the baby is born also, the liver will be severely affected, leads to the death of the fetus. So the entire thing I will repeat again. So here, when an Rh negative mother is marrying an Rh positive father, or a Rh negative woman is marrying a Rh positive man, the baby, if it is Rh positive, then in the first pregnancy, there is no harm. Even though the mother is exposed to that Rh antigen which the baby is having, the antibody in response to that antigen which is produced is of IgM type. That is anti-D which is produced in response, that is IgM type, which can't cross the placenta and destroys the fetus. So first baby is safe and even the mother is also safe. But in her next pregnancies, the mother already knows this RH, that is RH factor, that is RH antigen, and her body already have that, that is anti-D antibody. In frequent pregnancies, the antibodies which will be produced are of IgG type. So whenever this mother becomes pregnant for the second time, already her blood is exposed to that. Yes, after the birth of the first baby. So her body reacted to that and produced the IgM. But again, when the during the second pregnancy, again when it is exposed, I am telling that it is producing IgG type. So this IgG antibody is very small. It can cross the placenta also. This IgG is only the antibody which can cross the placenta. So this IgG antibody crosses the placenta and enters into the fetus. So when it is entering, what it is doing? This IgG antibody is clumping, that means destroying the blood cells, RBC of the fetus. And this RBC are still in the erythroblast stage. Means during the formation of RBC, there are several stages. That is reticulocyte stage, erythroblast side, then it is coming to erythrocyte stage, which we are calling it as RBC. So here, the RBCs are destroyed in the erythroblast stage itself. So we are calling it as erythroblastosis fetalis. So it is causing the death of the fetus by the destroying the erythroblasts. And even whenever even the birth happens also, it results in the death of the fetus. Why? Because the liver is severely damaged because of the release of bilirubirin and bilirubin. Okay, so this is about erythroblastosis fetalis. Are you clear? So here it is clearly explained. So the first born child is unaffected because the fetal and maternal circulation are separate, because the placenta didn't after the delivery only, the placenta, there will be the rupture of the placenta. That is even the maternal blood and the fetal blood will maternal blood and the fetal blood will be mixed only after the birth. Okay, after even though it is mixed also, the antibodies which are produced in response to that are of IgM type, and they will not cross the placenta, so it is safe. But during the second pregnancy, what happens? The mother's antibody the mother's blood is already sensitized, and she produces what type of antibodies? G type of antibodies, which immediately cross the placenta and destroys the that is RBC, that is erythroblast stage of the fetus. So we are calling it as erythroblastosis fetalis. So now You may have the doubt, then how can we prevent it, madam? Yes, we can prevent it. Now the technology, that is science, has improved very much. Now there is a vaccine. Okay, there is a vaccine which is called as Rogam vaccine. Wait, there is a vaccine which is called as Rogam vaccine. So, which forms a barrier like thing between the mother and the fetus, so that the antibodies of the mother, that is IgG antibodies, will not enter into the fetus. Means this Rogam flows out, has to coagulate or clot, what we will be calling it as coagulation of blood. When we see the coagulation of blood, so we have seen that platelets, when we studied about the components of the blood, so the platelets are mainly responsible for coagulation. These blood platelets, uh, which are present in the blood, are mainly responsible for the coagulation. So how they coagulate? So these platelets will arrange themselves at the tissue place where there is damage, and they produce some factors along with the substance which is called as thrombokinase. Okay, so they produce the substance which is called as thrombokinase. So this thrombokinase which is produced, what it do, it converts this inactive prothrombin into active thrombin, and this thrombin which is produced again converts fibrinogen into fibrin. This fibrin will be normally in the form of threads. Immediately when it becomes active, it forms like this. It forms a clot like thing, which we will be calling it as blood clot. So at the damaged site, what is happening? This thrombokinase, which is released from the platelets, is converting prothrombin into thrombin, and this thrombin is converting fibrinogen into fibrin. This fibrin forms a clot, which we are calling it as blood clot, preventing the further flow. Okay, this is about blood coagulation. So now coming to the another word, that is lymph. Usually we will be studying more when we study about apoptosis, which is the cell death. That is when the cell death doesn't occur, it accumulates in the lymphatic nodes, which is due to the metastasis, a type of the cancer, which is a famous research in one of our national institute, that is NCBS. Mm, Now we will study again. We'll come back to the topic, that is lymph. So what is lymph? So we know there are several type of blood cells, RBCs, WBCs, platelets. We'll draw with different colors. Okay, RBCs, WBCs, platelets, everything are moving through our capillaries. So likewise, all the children are moving in one road. But there are some naughty children. Instead of going to their home, they want to come out. Okay, they will be coming out of the way, and they miss the way also. Yes, they will miss the way, and they will be coming out. And these here we are studying about cells. Yes, these cells which came out of their normal path, where they missed the way, again all these will be collected again. Means normally the blood cells are moving in one row. Yes, all the blood cells are moving like this. It is, think this as capillary, and all the blood cells are moving like this. But some blood cells don't want to follow them. They will come out. They miss the way. They don't know how to go. They will be present in the tissue fluid. Later, all these also will be collected again in some capillaries, which we will be calling them as lymphatic capillaries. And they will be, everything will be accumulated at one place, which we are calling them as lymph. So the lymph is formed when some of the blood cells ooze out of the capillary wall, and they will be collected again in some vessels, which we are calling them as lymphatic vessels, which form the lymphatic capillaries and form deposits at one place, what we call them as lymph. Okay, so lymph is nothing but a tissue fluid. Okay, it is a tissue fluid. See here, the blood is flowing. Yes, some of the some of the cells will be oozing out, and they will be again collected in the lymphatic system, which we are calling them as lymph. Okay, so it acts as it acts as a middleman between the blood as well as tissue. Lymph, we call it as a middleman between the blood and the tissue. Yes, that's it for this video. So in this video, we have discussed the different blood groups present in the human beings, how to identify the blood groups, okay, how to identify the different blood groups, and even we have discussed about a little note on coagulation of blood as well as lymph. Okay, hope you understood this topic. So again, in the next video, I will be coming before you explaining you the structure of the heart. Hope you enjoyed this topic, understood this topic. If you understood, please give a like, share and subscribe my channel, Swarna Ma'am's Biology. Okay, until then, bye.